Thank you for your kind introduction. The title of my presentation is iPS cells-derived and patient-derived intestinal organoids for gastrointestinal research. For today's agenda, I will first give a brief overview of organoids, what they are, their advantages and limitations. Then I will introduce you to our iPS-derived colon organoid system and how to differentiate human iPS cells to colon organoids. I will also discuss the applications that can be used with these colon organoids. Lastly, I will go over our patient-derived intestinal organoid biobank. So what are organoids? Organoids are self-organizing three-dimensional structures derived from either pluripotent stem cells or from primary tissue with the ability to recapitulate some of the spatial architecture and in vivo function of organs. Recent years, 3D organoids have emerged as an important tool for disease modeling. Over the last several years, the number of scientific publications utilize organoids has grown exponentially. Organoids are different from spheroid culture in that spheroids are 3D cultures derived from established monolayer cell lines, and thus may lack the complex cellular heterogeneity and architecture of the tissue of origin. There are advantages of using organoids because they are derived from patient tissues or stem cells. They contain mixture of cell type and thus mimic the complex in vivo cellular heterogeneity of the primary organ. They can be frozen and expanded over multiple passages. Biobanks may be created from them. Finally, they can be genetically manipulated using CRISPR technology. There are also some limitations for organoids culture as well. Isolation and culture of organoids from primary tissues may require experience and training, while protocols to differentiate iPS cells to organoids are lengthy and complex. They also require expensive factors such as osbonding, Win3A, and noggin for culturing. These factors may require lot-to-lot qualifications to reduce the variability between lots. Organoids can also be variable shape and size, thus making it difficult to implement them in high-throughput screening assays. Typically, organoids are derived from either embryonic or induced pluripotent stem cells or from adult stem cells resected or biopsied from organs. Both types have the unique capacity to self-renew and differentiate, which are the defining feature of stem cells. The left schematic shows the generation of an adult stem cell derived, also known as patient-derived organoid, from primary tissue biopsies. Tissues are dissociated into single-cell suspension and then embedded in an extracellular matrix, such as matrigel, and grown in the presence of specific growth factors to direct cell differentiation. These specific growth factors may vary depending on the tissue being cultured. The right schematic depicts the generation of a pluripotent stem cells or iPS-derived organoids from 2D stem cells cultures that mature into spheroids. These spheroids may be embedded in an extracellular matrix and grown in the presence of specific growth factors to the tissue of interest. To circumvent some of the limitations mentioned earlier, Sigma has developed our human colon organoid system. The system includes kits to allow researchers to take the human iPS cells and through a stepwise process differentiate pluripotent stem cells to definitive endoderm, and then to Hindgut Endoderm, and eventually into colon organoids. The entire process takes around 48 days. The media kits are chemically defined. They do not contain serum and are subject to stringent quality control. Our differentiation media kits have been validated on 2 human iPS lines, one from human foreskin fibroblasts and the other from PBMC-derived human iPS cells. We show that the resulting colon organoids express appropriate markers indicative of multiple cell types found in the colon, including goblet cells, enterocytes, Pennant cells, and enteroendocrine cells. Our colon organoids can be cryopreserved, propagated, and expanded over multiple passages. And for researchers who may not want to go through the lengthy differentiation process, we also offer cryopreserved human iPS-derived colon organoid and the expansion media. The colon organoids can be used for a variety of applications, including GI absorption and toxicity screens of oral drugs, metabolism, infectious disease modeling, and microbiome research. Step 1 is to start with a high-quality human iPS cells grown in feeder-free conditions. If your iPS cells are not already in feeder-free conditions, we recommend that you transition them to feeder-free culture for several passages before starting. iPS cells should express high level of pluripotent markers such as OCT4, 12-1-60, and SC-4. Make sure your iPS cells are undifferentiated and by having compact colonies with distinct borders and well-defined edges as shown. Differentiated cells must be cleaned off under a dissecting microscope before initiation of differentiation. Once your iPS cells have been determined to be of high quality, start dissociate iPS cells to single cells using Accumax and culture overnight in the presence of ROCK inhibitors. The next day, cells should be about approximately 85 to 90% confluent. Start definitive endoderm differentiation by adding definitive endoderm induction media from day 1 to day 3. It's a 3-day induction process. Change media every 24 hours. On day 4, one of the wells is sacrificed for co-staining with CXCL4 and SOC17 to test for the differentiation efficiency for definitive endoderm by flow cytometry. Double positive for CXCL4 plus SOC17 must be greater than 90% before proceeding to the hindgut endoderm differentiation. To analyze by flow for the percent purity of your definitive endoderm, start with an unstained sample and create quadrant gate with the unstained sample. Then use this gate to determine the percent positive of CXCL4 alone, and then SOC17 alone, and most importantly, the CXCL4 plus SOC17 double positive. As you can see, the percentage purity of CXCL4 plus SOC17 population is greater than 90%. If for some reason the percent purity of CXCL4 plus SOC17 double positive is below 90%, then the possible causes are your starting iPS cells are not of high quality. We recommend to check for pluripotency for OCT4, 12-1-60, and the SC-4 markers. Also make sure that your iPS cells are pure and undifferentiated before initiating differentiation. Also, your iPS cells on day 1 is either too sparse or too confluent. Make sure your iPS cells are completely single cell dissociated before counting, because clumps can affect accuracy in cell count. On day 4 to day 7, begin hindgut endoderm differentiation by adding hindgut endoderm induction media to the culture every 24 hours. On day 8, sacrifice 1 well for the flow analysis for the expression of the hindgut endoderm marker CDX2 before proceeding. Percent CDX2 positive must be greater than 60%. The image on the lower left shows the appearance of spheroids on day 6 of differentiation. Spheroids are more abundant on day 7 and day 8. The dot plot on the right is an example of our flow analysis for the expression of CDX2 on day 8 of differentiation. The higher the percentage of CDX2, the better. If the percentage of CDX2 is below 60, the possible causes are, again, your iPS cells are not of high quality, and also your iPS cells on day 1 is either too sparse or too confluent. On day 8, transitioned to 3D grow human colon expansion media. Change media every other day until day 20. On day 20, dissociate into single cells and embed in 25 microliter growth factor reduced matrigel dome. We recommend doing a 1:50 and 1:100 dilutions first to determine the optimal number of cells to embed. Aim for approximately 90% occupancy in each dome. The image on the lower left shows the appearance of hindgut spheroids on day 12 of differentiation. More hindgut spheroids are present on day 20. An example image is shown of single cells from day 20 embedded into 25 microliter growth factor-reduced major gel dome in 24-well plate. The appearance of colon organoids with buddings are observed on day 12 in the dome as shown on the image to the right. Our iPS-derived colon organoids can be passaged over a long period of time. The image on the left is the first 12 days in growth factor-reduced major gel dome designated as passage 0. Note that the developing organoids are not mature yet, but they do have much budding. But they do not have much budding. Day 42 is day 10 of passage 1. Organoids are bigger and have multiple buddings. Day 52 is day 10 of passage 2, and organoids have complex structures and budding. At this stage, these organoids are considered to be mature. Day 428 is day 9 of passage 37, and it serves as an example that our iPS-derived colon organoids can be passaged at many times. The organoids still have the complex 2D structures and budding. We recommend passage every 10 to 12 days using organoid dissociation reagent. Do not exceed 12 days before passaging. On the first 2 days of every passage, ROCK inhibitor is added to enhance viability. Thereafter, ROCK inhibitor is not necessary. Organoids can be cryopreserved in colon expansion media plus 10% DMSO or organoid freeze media. It is important to monitor your organoid culture daily. As you can see, the organoid looks translucent on day 10 and day 12. From day 14 onward, organoid looks less translucent and centers look very dark and dense. Thus, we determined that the optimal passage time is 10 to 12 days, preferably 10 days. If you go past 14 days, the organoids start to form dark necrotic balls that start to disintegrate at the edges as shown by the red arrows. For those that do not prefer to go through the differentiation protocol, we also offer cryopreserved human iPS-derived colon organoids and expansion media. Our cryopreserved 3D grown human colon organoids contain about 2,400 organoids per vial, sufficient for 12 times 25 microliter dome in 24-well plate. Each dome has about 60 to 70% organoid occupancy 2 days after thaw, as seen on the image to the left. By day 7, there are about 90% organoids in each dome. We recommend passaging at conservative 1:3 ratio until become more familiar with culturing organoids. If by day 10 you don't see 90% occupancy, you can still salvage by passaging at a more conservative 1:2 ratio. Here are some examples and recommendations. From 70 to 90% occupancy, passage at 1:3 until become more familiar with culturing organoids. From 40 to 70% occupancy, passage at 1:2. From 30 and below, recede back into the same number of domes. We characterize our iPS-derived colon organoids with these markers on the list by whole mount immunofluorescence staining. The 4 that are highlighted in red are the colon-specific markers. They are carbonic anhydrous 2, carbonic anhydrous 4, mucin 5B, and SAB-B2. And they are all expressed in our iPS-derived colon organoids in addition to the rest on the list. Here are the ICC data of the stainings. They are positive for the colon-specific markers, calcium carbonic anhydrous 2, carbonic anhydrous 4, and mucin 5B. The posterior hindgut celiac 2 marker is expressed along with LGR5, which is the stem cell marker expressed at the base of the crypt. Also positive are EPHB2, SCA1, mucin 2, E-cadherin, which is an epithelial marker, and TPH-1, an enteroendocrine cell marker. To confirm the expression of GATA4 and SAB-B2 of our iPS-derived colon organoids, we stained alongside a patient-derived colon organoid line and found that both expresses SAB-B2 and GATA4. Both markers are expressed in the nucleus and they overlay with DAB. We found that GATA4 mRNA is expressed in the stomach, duodenum, small intestine, and colon from both sequencing data on proteinatlas.org as shown here. We get asked often whether our iPS-derived colon organoids express vimentin, which is a mesenchymal cell marker. We tested for the expression of vimentin by flow cytometry using our human iPS cells and MCF7 cells as a negative controls, and MDA-MB231 as positive control. Results show that our iPS-derived colon organoids are negative for vimentin. We recently sequenced our human iPS-derived colon organoid and a patient-derived colon organoid line along with our iPS cells by RNA-seq. Here is a bi-clustering heat map that shows the expressing profile of the top 30 differentially expressed genes in both iPS-derived and patient-derived colon organoids. Their expression patterns are very similar to each other, but are very different from the iPS cells. Besides the top 3 differentially expressed genes, both iPS-derived colon organoids and patient-derived colon organoids share similar expression trends of some of the colon genes as shown in this graph. Next, I want to shift our attention to the cytotoxicity drug screen application data using our human iPS-derived colon organoids. For the cytotoxicity drug screen, 7 compounds with well-known effects along with 3 Merck Serono compounds were tested. Screening was performed on 4 days old organoid. 4 replicates were performed with 8 concentration ranging from 10 nanomolar to 300 micromolar. DMSO was used as a vehicle control. Metformin, a drug used to treat type 2 diabetes, which is known to be well tolerated, was used as a control as well. Cell viability was assayed after 72 hours of treatment. Protocol is fairly simple and is also detailed on our Sigma website. So basically, harvest domes and passage according to protocol. Feed 10 microliter matrigel domes into 96 well plates in 100 microliter expansion media plus ROCK inhibitor. Incubate at 37 degrees Celsius in the presence of 5% CO2 for 3 days. On day 4, replace the media with the compound, and then measure cell viability at 72 hours later. These are the dose-response cytotoxicity results of different drugs on our iPS-derived colon organoid. Flavopiridol was the first CDK inhibitor in human clinical trials. It is known to induce programmed cell death. Its toxic effect on our iPS-derived colon organoids is consistent with expectation. Gefitinib and loperamide also exhibit toxic effect as expected. For metformin, we do not observe any apparent cytotoxicity even at 750 micromolar. This is consistent with expectation. To the left are examples of drugs which have no apparent cytotoxicity effects on our colon organoid. They are paracetamol, Tranen, Tylenol, and ketoprofen, a non-steroidal anti-inflammatory drug that is used to treat arthritis, and alosetron, Tranen Lotronex, which is used to treat diarrhea and severe irritable bowel syndrome. Of the drug tested, 7 of 9 compounds confirmed with expectations. Ongoing quantifications of additional GI tox compounds is required to determine predictive value of this GI tox model. For high throughput screens, it is important to have consistent well-to-well reproducibility. In this experiment, we dissociate colon organoids into single cells and plated them at 25,000 cells per dome. 5 days after seeding, we see that each well contains dome with similar number of viable cells. Single cells associated colon organoids will reassemble into 3D organoids as shown in the image on the right. There is size variability within wells, but well-to-well consistency observed. Different plating density are possible, thus providing flexibility in when to initiate experimental treatment. We also performed forskolin-induced swelling assay on our iPS-derived colon organoids and found that the cystic fibrosis transmembrane conductance regulator genes is functional. The iPS-derived colon organoids in the left image do not swell in the absence of forskolin. When 10 microliter of forskolin is added to the culture, swelling is observed starting at 3.5 hours and onward. A 36-hour time lapse of this is available on our website. We now offer a comprehensive biobank of highly characterized tissue-derived human gastrointestinal organoids from normal and diseased patients, which have been developed by researchers at the University of Michigan Center for Gastrointestinal Research. There will be a total of 58 PDOs derived from the stomach, the duodenum, the ileum of the small intestine, colon, and rectal samples, 17 of which has recently launched, and RNA-seq data for each is available. Normal and disease derived from colon adenoma, ulcerative colitis, and Crohn's disease are also on the list. These PDOs are of low passage number and highly viable upon thaw. All are cultured in our optimized 3D grow LWRN conditioned media. Here are the images of a few of our PDOs. 5 are from normal tissues, and the one on the lower right-hand corner is from colorectal adenoma tissues. Here is a detailed list from our webpage showing the age, sex, organ, and whether the PDOs are from normal or diseased tissues. The first 17 in blue, again, are recently launched. Here is the characterization data by ICC of a patient-derived colon organoid line. It's positive for colon-specific markers, the carbonic anhydrase 2, carbonic anhydrase 4, and mucin 5B. The posterior hindgut celiac2 marker is expressed along with LRG5, which is the stem cell marker expressed at the base of the crypt. It is also possible for SCA1, E-cadherin, and epithelial marker, and TPH-1, and enteroendocrine cell marker. Now I would like to briefly discuss the SARS-CoV-2 and its effect on the gut enterocytes. Recent publications have shown that the intestines may present another viral target organ. Studies have revealed that SARS-CoV-2 receptor angiotensin converting enzyme 2, ACE2 for short, is highly expressed in the small intestines. Let's first look at the general structure of SARS-CoV coronavirus. Figure 1A depicts the structure of SARS-CoV-2 virion with the main set of glycoproteins. Of the 4 essential glycoproteins, it is the S, or the spike glycoprotein, that binds to the host cell's A2 receptors that allow for viral entry. Figures 1B show a linear map of the spike glycoprotein. Its N-terminus S1 contains the receptor binding domain, which is required for binding of the host ACE2 receptor. The C-terminus S2 unit contains a functional element required for fusion. It also contains a transmembrane domain for membrane anchoring and a cytoplasmic tail for appropriate intracellular trafficking. There are 2 routes of viral entry into the host cells. One is by receptor-mediated endocytosis initiated by the binding of the viral spike glycoprotein to the ACE2 receptor on the host cells. The virus then ends up in the endosome, where the low pH level activates cathepsin L. Cathepsin L then activates the viral spike glycoprotein and facilitates viral membrane fusion, resulting in the release of its genetic material out of the endosome and into the cytoplasm. Alternatively, the spike glycoprotein can be cleaved by the surface protease on the host cells called TMPRSS2, and this can lead to the direct fusion of the viral membrane and the plasma membrane, resulting in the release of the viral genetic material into the cytoplasm. Our patient-derived duodenum organoid expresses both ACE2 and TMPRSS2 markers. Thus, it may be suitable for use as an experimental model for the study of coronavirus infection and its biology. Besides our human intestinal organoid, we also offer general organoid qualified reagents such as our LWRN conditioned media supplement for culturing PDOs, our all-responding 1 conditioned media supplement. We also offer organoid dissociation reagent and our organoid freezing medium. We also have our all-responding 1 expression 293T cell line available for making your own conditioned media. We recently launched our human lung organoid system as shown here. The system consists of all of the necessary differentiation media for generating mature lung bud organoids. We also have our human iPS-derived anterior foregut endoderm available for further differentiation to mature lung bud organoids. In summary, human colon organoids can be generated from human iPS cells using our stepwise differentiation protocol, along with our chemically defined serum-free media kit. Our iPS-derived colon organoids can be cryopreserved, propagated, and expanded in long-term culture. They express appropriate markers indicative of multiple cell types found in the colon. They can be single-cell dissociated to control for consistency in organoid numbers from well to well for high-throughput screening. They show similar trends to that of patient-derived colon organoids by RNA-seq data. They can be used in cytotoxicity drug screening. Also, the cystic fibrosis transmembrane conducting regulator gene is functional. 58 total gastrointestinal organoids are available from normal and diseased tissues with low passage number. Our patient-derived duodenum organoids express the 2 SARS-CoV-2 cellular entry markers ACE2 and TMPRSS2. Lastly, I would like to acknowledge my colleagues on the list for making the 3D organoid products possible. I also would like to recognize our Merck Healthcare in Germany for the cytotoxicity data. Lastly, I would like to recognize the team at the University of Michigan Center for Gastrointestinal Research for making the PDO Biobank possible. Thank you for your attention, and I'm looking forward to your questions.